Hi everyone! Today's lecture is going to be about DNA mutations and DNA repair proteins. I couldn't resist but put up an image of the X-Men, who are all supposed to be mutants, and the mutations in their DNA allow them to have such amazing powers as shooting laser beams out of one's eyes, flying, or going through walls. And unfortunately, most DNA mutations are a lot more prosaic. But what I wanted to tell you guys, and the reason I put up this picture, is because all of you listening to this lecture have DNA mutations in your cells as well. Now, unfortunately, they do not give us the ability to fly, but they still can create all kinds of problems, especially the ability for cells to turn from normal cells to cancerous cells and stop listening to the instructions of the body over-proliferate and cause our demise. As a result, the topic of DNA repair is a very important topic in molecular biology, because if we can understand DNA repair mechanisms in our cells, we can help find out various ways of preventing cancer or combating cancer. Now, what are DNA mutations? A DNA mutation is not merely a mistake that happened after DNA replication, but it's a permanent change in the DNA sequence that can be inherited. So, in this image here, a mistake that occurred right here can still be fixed by a DNA repair mechanism. But if the DNA repair mechanism neglected fixing this incorrect base pairing from where a T was changed to a C, and the cell replicates again and uses this DNA strand as a template, the mistake is permanently fixed in the daughter cells. It will persist there, and the DNA repair machinery has no way of finding it. So it's very important for the DNA repair machinery to recognize these sort of errors that occur periodically inside of our DNA and catch them before the next round of DNA replication can occur. As a result, if you guys remember cell cycle, and the fact that cell cycle is made out of 4 parts, the G1, the S phase, the G2, and the M phase when the cell actually divides. A big part of preparation for DNA replication in the S phase, so the G1 phase, it prepares the cell for replication of its own DNA, is making sure the DNA template that will be used for replication is free of errors. And all of these checkpoints have to be passed before the cell clears, permits the DNA to be replicated. DNA mutations can be bad, good, or of no consequence. They can be good and, for example, make one particular organism of the species more likely to resist certain environmental hazards. They can be bad and result in the organism's death. Or most mutations actually have no consequence on the cell because of many reasons that we will discuss as the class progresses. Several reasons I can bring up right now is that our genetic code is degenerate. So several different nucleotide combinations can code for the same amino acid. The other important thing for eukaryotes is that a lot of the DNA that eukaryotes carry is not actually coding for anything. So if a mutation happened in that particular location, it might affect whether or not an intron, it might not affect how a protein is made. However, even certain single nucleotide mutations in a particular coding region of the protein that, for example, cause an amino acid to be then read as a stop, can have major effects on an organism, because now the protein that was produced earlier is basically knocked out and is no longer produced. DNA repair, so one important thing to take away from today's lecture is that DNA repair proteins do not repair mutations. By the time there is a mutation like that in the DNA, there's no way a DNA repair mechanism can recognize it. However, DNA repair occurs earlier, and it scans the DNA for any sort of aberration and fixes these errors, bumps, nicks, and so forth before the next cycle of DNA replication can occur. So here, there's still a chance of repairing the mutation in this phase of the error, when the error just occurred, but there's no longer a chance to repair the mutation here because this GNC base pairing looks correct. The daughter strand and the mother strand match. There's nothing out of ordinary in this G and C base pairing that the DNA repair mechanism can recognize. So now I would like to talk about the different types of mutations that occur in the DNA. There's two main big types of mutations the DNA repair proteins deal with, that DNA repair proteins prevent. There's point mutations, and there's the indel mutations. Indel is a portmanteau of insertion and deletion, and it's a word very frequently used in evolutionary biology and in genetics, so I want you guys to know it. So an indel is both an insertion and deletion, because both insertion and deletions have very similar effect on the protein, on the DNA structure, especially on protein coding. So back to mutations. As I said, there are point mutations and indels. So let's now talk about different types of point mutations. The most common type of point mutations are silent mutations that result, even though they resulted in a permanent change in the DNA, a base was changed to a different base, it does not change the protein coding because our genetic code is degenerate, and not every base pair substitution change will result in actual change inside of the amino acid that is used. Another type of point mutations are missense mutations. For example, due to the change on the nucleotide level, now instead of a tyrosine, the ribosome will place a proline in that particular location of the protein because the amino acid the nucleotide sequence no longer codes for tyrosine but codes for proline. And this is how these missense mutations are frequently notated. It's a typical way to write out different point mutations, and a good reason to learn single letter amino acid abbreviations in the future for you guys. So Y is tyrosine and P is proline, and 214 is the amino acid position of this tyrosine that then was changed to a proline. It was amino acids 214 inside that particular stretch of the polypeptide. Finally, another typical point mutations are nonsense mutations, aka mutations that will result in a stop instead of another amino acid. That truncates the protein at that amino acid location, and the nonsense mutation is denoted as an X, stop. So while missense mutations can change protein structure and, for example, cause the unfolding of a protein at the worst, or change the active site of an enzyme or binding site of a receptor, nonsense mutations are much more dramatic because they will result in practically no protein produced. Or, for example, a protein that has the catalytic domain but is lacking a regulatory domain and can no longer be controlled by the cell, which is very bad. Now, indel mutations also come in several flavors too. The worst of them are the frame shift mutations that result in insertion or deletion of nucleotides that did not come in multiples of 3. Why is that? Well, our genetic code uses 3 nucleotides to get 1 amino acid. So if 4 nucleotides are removed, the genetic code has been shifted, and the rest of the mRNA that is translated into a protein will no longer produce any of the correct amino acids. Now, there's also indels that will result in insertion or removal of of nucleotides in multiples of 3. They're far less traumatic and commonly found in our DNA, both inside proteins that can cause some proteins to become fragile if these stretches grow too long, or these types of insertion and deletion mutation, these indel mutations, can also be used, for example, in the short tandem repeat DNA analysis that we talked about last week. So you can use these growing or shrinking indel stretches of DNA to track down individuals and figure out which individuals are related to whom. So how can point mutations happen? Point mutations can happen due to damaged bases in the template that were damaged due to UV radiation, chemical exposure, reactive oxygen species, even water can damage DNA. Or they can be due to DNA polymerase mistakes that weren't caught and repaired in time before the next replication occurred. Indels can also happen. During DNA replication due to DNA template slippage. They can happen due to aberrant recombination events during cell division, or they can happen, and I didn't write it down, when double strand breaks in DNA occur and they're repaired imprecisely. So DNA repair mechanism can actually cause mutations when they're trying to do a good thing. And then another type of mutations that your book describes, and I will not go over today at all, are chromosomal abnormalities due to either double-strand breaks that were repaired imprecisely, or aberrant recombination events, or other errors during cell division. So a very famous mutation that is due to chromosomal abnormality mutations is Down syndrome. Some types of Down syndrome, you will get an extra chromosome 23 or just a chunk of that chromosome attached to chromosome 20, attached to different chromosome, which results in production of overproduction of certain proteins and the Down syndrome phenotype. So a lot of the DNA repair mechanisms we'll discuss are used to fix point mutations, and let's focus on them first. This slide is a diagram that I got of the R&D Systems website, and it's not part of your book, but summarizes nicely the different all the various dangers that our DNA faces on a regular basis. So cellular metabolism will cause damage to the DNA. So that's reactive oxygen species that are produced due to cellular respiration by our mitochondria. Now, our cells have a lot of ways to deactivate these reactive oxygen species, but they still can damage the DNA, and the damage needs to be repaired. The sun, or UV radiation, will cause thymidine dimers that have to be excised and fixed. And if this mechanism is not working precisely, human patients can develop various skin conditions, as well as cancer. So another type of insult to the DNA are X-rays or radiation. So all kinds of types of ionizing radiation. And chemical exposure. So chemical exposure can be carcinogens or nitrates that we consume in our food, but it can also be the molecules commonly found all around the DNA, such as water. All of these, if a chemical can interact with the DNA and change it, it is a potential mutagen. Finally, DNA polymerase, as beautiful as it is, as precise as it is, does make mistakes, and those mistakes have also need to be repaired. So here we go. We have the various outcomes of DNA damage, and the only one we're going to talk about today is DNA repair. So how do cells prevent mutations to DNA? One of the ways, so there's 3 typical ways. So you can get mutations before and after DNA replication, which need to be reversed before DNA is replicated. During DNA replication, both the DNA polymerase has proofreading ability, and there's another set of proofreading enzymes that follows right after it. And If large mistakes occur, the other side of the chromosome, homologous chromosomes can be used as template to fix large errors. This is a laundry list from your book showing that the different examples of DNA damage. Due to time constraints, we will not go over all of them in lecture, and I will not test you on all of them, but you should know at least the ones that I'm covering in lecture today. Where today we're going to talk about mismatch repair and double-strand breaks, and then abasic sites, such as deaminated bases. And for all of these different types of DNA damage, there's different types of repair processes that involve multiple enzymes that are used. And your book specifically mentions that DNA repair mechanisms are among the most energy costly in the cell. They use a lot of ATP, but it is such an essential function for the cell to make sure that its DNA is intact and can be faithfully passed on onto the next generation, that the cell readily expends a lot of energy for the various DNA repair processes. And now this part of the figure just shows the important enzymes, here in this case both eukaryotic and prokaryotic, that play a role in these various processes, such as exonucleases in nucleotide excision repair. So right now we're going to talk how DNA repair recognizes and fixes base pair errors before and after DNA replication. And a lot of these base pair errors are can be caused by chemical exposure. And in this case, we're going to discuss the evils of water and what water, which is a charged dipole and has plenty of chemical potential, can do to our DNA. So water is capable of participating in deamination reactions, aka any reaction where the water molecule reacts with an amino group and makes it and replaces the amino group, making it leave as ammonia. So in this case, if this deamination reaction happens occurs with a cytosine base, it will change the cytosine base to a uracil, because the only difference between a uracil and a cytosine is that the cytosine has an amino group and uracil has an oxygen group here. So because a deamination has occurred, the deamination reaction causes a chemical change to the base. Cytosine is no longer a cytosine, but it's a uracil. And the outcome of this reaction is that the genetic code of the DNA has been changed.